Okay, good afternoon to everybody. So on behalf of the two other organizers of the science class Colloquio, Augusto Sagnotti and Chiara Cappelli, they apologize, they cannot be here with us today. I'm happy to welcome you to today's colloquium. So this is a rather special month for many reasons. One reason concerning the colloquia is that we will actually have two science class colloquia in this month. There will be another one next week. And actually it will also take place at an unusual time. It will be at 2 p.m. instead of 3 p.m. But anyhow, this is just a communication. I mean, concerning today's colloquium, the speaker is Professor Arthur Lesk from Penn State University. He will speak about the architecture of proteins, but let me introduce Professor Catania, who will actually tell us a little bit more about the speaker and the topic of today. Okay, thank you, and thank you for coming.
Good afternoon to everyone. It's a pleasure and honor to have today with us Arthur Lesk. He's professor of biochemistry and molecular biology at Penn State University. Before this, he was for many years associated to Cambridge University and also with the MRC Laboratory of Molecular Biology in Cambridge. Arthur is one of the fathers and founders of bioinformatics, and he has given and provided seminal contributions to the field besides starting the field. And I will only mention a few of his achievements, also on the suggestion by Arthur that I should keep this introduction brief. And he and Scienze discovered the relationship between amino acid sequences changes and changes in protein structure. And this is an insight and the method and results that have provided the basis of a fundamental method of homology modeling that is a crucial method with which structures of proteins are studied. Also in association with Cyrus Scienze, Arthur studied the conformations of the antigen binding sites of antibodies. They discovered the so-called canonical structures of complementarity determining regions of the antibodies, that are the regions of antibodies with which that make contact with the antigen. And this has provided a theoretical basis for the widely used method of humanization of therapeutic antibodies. And we have now in medical practice antibody drugs derived by these methods. And he wrote the first computer programs to be used to describe and to illustrate structures of proteins, and he derived algorithms that are widely used in the community to analyze the structure of proteins. And these are only a few of his many achievements with which Arthur provided the scientific community with fundamental insights into how to use computational and bioinformatic tools to get insights into the structure of proteins. In addition to his great scientific achievements, Arthur is the author of very popular must-have books on bioinformatics and genomics. And today he will talk on the architecture of proteins, and he will describe to us about his efforts towards deriving an equivalent of a periodic table with the fundamental protein folding elements that shape the structure of proteins. And last, I would like to remind you that this, as a colloquium, will be a very general talk that will be understandable also for people outside of the field. But Arthur will give a series of lectures that are, the advert has been circulated, and we shall circulate again this advert in the next. These lectures will take place in the next couple of weeks, and everyone is more than welcome to attend these lectures. So, Arthur, thank you very much.
Thank you. It's a great pleasure to be here. I thank the organizers for the invitation and the previous speaker for the very generous introduction. I'm going to talk first with some general background, which probably many of you already know, and then if there's time, go on to a particular research project in which I've been involved. So the basic idea is embodied in the central dogma of molecular biology, which was stated by Francis Crick. DNA, can I use this as a pointer? No. DNA contains the information repository. DNA makes RNA, makes protein. Proteins are the executive branch of the cell. So the structure of phosphofructokinase, which is a typical enzyme, contains thousands and thousands of atoms. You can see within the picture there are a few small molecules represented by spheres, and what the enzyme is doing is holding two of these together so that the reaction can take place. So enzymes are the executive branch. How this works, so you have DNA as the information repository, double-stranded, which gives an easy mechanism for replication. In addition, DNA is transcribed to RNA, which is translated into proteins, and that's where we get the proteins from. When Francis Crick originally stated the central dogma, he emphasized the one direction of information transfer, DNA to RNA to protein. What he didn't know at the time, but which we've since learned, is that certain viruses can achieve reverse transcription. They can take RNA and put it back into DNA. But that's a correction, not an important one for today's topic. So proteins have many functions. Some of them are structural, some of them are catalytic, like phosphofructokinase. There are obviously proteins that interact with DNA. Regulatory proteins, very important. Proteins don't just do things, but they organize how everything else is achieved in the cell. Antibodies, and the structure on the lower right is an IgG, and transport and storage proteins, of which hemoglobin is perhaps the best known. The structure at the top is sperm whale myoglobin. It was the first protein structure solved by X-ray crystallography, and it is the recipient of the oxygen transferred by hemoglobin from the lungs to the muscles.
But it doesn't show well on that screen, but anyway, better than
Okay, well. Now each function requires a different protein structure. So whereas DNA has basically one structure, that's not quite true, but it's very close to true. But we have many, many protein structures. And the Worldwide Protein Data Bank has hundreds of thousands of entries. One is in the position that there's a statement attributed to Charles de Gaulle, though he probably never said it. How can anyone govern a nation that has 246 different kinds of cheese? So if you have all these proteins, how can you make sense of them? Another quote is that Rutherford said that all science is either physics or stamp collecting. And my response to that is that protein structure combines the best aspects of both. Because we do have many, many proteins, like the different kinds of stamps, but the hope is that we will find unifying principles the way the physicists do. Indeed, there is no question that proteins obey the laws of physics. But what we see in cells is a combination of physics and basically historical accident. And it can be very difficult to sort out what's being governed by the physics and what isn't necessarily true but is simply history. So if we see two proteins that share similarities, why is this? Is it because they're related by evolution, like Darwin's finches, or is it because physics wouldn't allow them to be any different from what we see? So the physics is there. It tells us what can or can't happen, but we have to appeal to history to find out what really did happen. What are the challenges? How does DNA sequence determine protein structure? And from protein structure, protein function, and the organization of cellular activity. Well, proteins are linear polymers of amino acids. DNA is a linear sequence of nucleotides. So those are both one-dimensional. Transcription takes DNA to RNA, translation takes RNA to amino acid sequence. amino acid sequences are still one-dimensional, but protein folding spontaneously takes the amino acid sequence and creates the three-dimensional structure. And this is the point, protein folding process, is the point at which nature makes this fantastic leap from one dimension to three dimensions. The next question, of course, is how does protein structure determine protein function? And the answer there is interactions. And that's a challenge because typically what the protein data bank entries give us, typically what a crystal structure or NMR structure or even electron microscopy structure determines is an individual protein. And to figure out what's going on, you have to figure out how they interact with things. So the picture shows a protein interacting with DNA. And what that protein is doing is controlling whether the gene in that region is expressed or not expressed. All our cells contain, with a few exceptions, all our cells contain the same DNA sequence. But our liver cells express a certain set of genes, our brain cells a certain, so there have to be control mechanisms despite the unique DNA sequence that controls what's going on. Now, what's happening, if we look a little more closely, the DNA sequence contains in the set of bases the code for the amino acid sequence. So it's read 3 bases at a time, and there's a simple translation table that tells you what amino acid corresponds to each set of 3 bases. And that is synthesized by the ribosome into an amino acid sequence, which folds spontaneously. Now, that is what Gilbert and Sullivan called the most ingenious paradox. And if you think about it, the translation to amino acid sequence is logically very simple. I could give you this slide and write down a nucleotide sequence and say, just take pencil and paper and figure out the amino acid sequence. You'd all be able to do it. I mean, take someone off the street, they'd be able to do it. Very simple logically. But the machinery required to achieve that involves the ribosome and many other auxiliary components. Very, very complicated machinery. Conversely, and this is what's paradoxical, folding is logically very complicated. If I gave you an amino acid sequence and said, what's the structure? Certainly without access to a computer, you'd be lost. But, although it's logically very complicated, it occurs spontaneously. So it turns out that all the information required to specify the protein structure is contained in the amino acid sequence, and it doesn't require any cellular machinery. You can do it with an isolated protein in a test tube. So then you could challenge us, well, couldn't you, if all the information to describe a protein structure is contained in the amino acid sequence, couldn't you write a computer program that would take an amino acid sequence and create as output the protein structure? In the last few years, there have been immense strides, you could even call them breakthroughs, in being able to do just that. So this is a picture of such a prediction. There is a program called CASP, Critical Assessment of Structure Prediction, that organizes blind tests. This is a prediction by one of the best groups in the field, David Baker, the University of Washington. And it's really spot on. One of the structures is experimental and the other is the prediction. And really, it's very, very good. The prediction is of comparable quality to the experimental structure determination. In fact, you can go on beyond this and not simply take natural amino acid sequences and predict their structures. You can design proteins. So if you wanted a catalyst for a reaction that was unknown in nature, there have been examples where people have designed amino acid sequences that not only folded up into what they expected, but folded up into a protein that had catalytic activity of the kind wanted. So molecular biologists used to be like astronomers in that we could observe the subjects of our interest but not do anything with them. But now there's a lot of control that can be done. So we're back to the varieties of cheese problem. There are many different protein structures. Here are a few of them. And what do? Well, we want to ask, what do they have in common? And if we were to take the time to stare at these pictures enough, you would see that, I don't even know how this worked. Well, what you would see, let me just go on. There is a substructure called a helix, which, if you look hard, you can see in several of these structures. So my problem is I can't see the screen very well. So that's a helix, and that's a helix in another molecule. So you begin to see that there are common substructures that recur in many proteins. And these secondary structures, which most of you know, is the alpha helix, which is formed from a local region of the chain. It's held together by hydrogen bonding, which is depicted in this diagram by broken lines. And as I say, that's of a purely local region of the chain. Then there are beta sheets in which several regions of the chain, which might very well not be consecutive in sequence, may be very distant in the sequence, are organized in space to interact laterally, again by hydrogen bonds. Now in these pictures, the chevrons show you the direction of the chain. So if you look closely, you can see on the upper right that all the strands are parallel. They're both all pointing up or down, I can't see which. And that gives rise to a characteristic hydrogen bonding pattern. In the lower right, the strands alternate up, down, up, down, up, down. That's called an antiparallel beta sheet. And you can see it gives rise to a different pattern of hydrogen bonding. You can also have mixed sheets, but these are the two extremes. In the lower beta sheet, there is one example of two consecutive strands in the sheet next to each other, linked by a short loop. And that's called a beta hairpin. So now we begin to build up higher levels of structure, which are called supersecondary structures. So supersecondary structures are recurrent patterns formed by consecutive secondary structure elements. So in the center, there's the beta hairpin, which is the same as the center of the lower right. Oops, beta hairpin. You can have an alpha hairpin, the helix hairpin, and in both of those cases, the chain's going up, across, and then back down. Another supersecondary structure is the beta alpha beta structure, which has a stranded beta sheet followed by a helix going the other way, followed by another strand of beta sheet that interacts laterally with the first one. So in that case, the two strands are parallel, not anti-parallel. Now, it's already been clear that a lot of what we do depends on being able to draw these pictures. And there's a cottage industry on developing representations. The left is a complete structure showing all non-hydrogen atoms in their full size. The center one is what's called a ball and stick representation, and the right is one of these cartoons. Now, the one on the right is perhaps the clearest because you can see the helices, you can see the strands of sheet. In fact, they're even made easy to see by being in different colors. But in some sense, such pictures are misleading, because it looks as if you could take a water molecule and just shoot it through the molecule, shoot it through the protein, because there are lots of holes there. But that's not true. The side chains aren't shown, and the point of the picture on the left is that it's clear that the whole thing is a compact array of atoms. If you were to shoot a water molecule at it, it would just bounce off. On the other hand, it would be very difficult, even for an expert, to see the alpha helices or strands of sheet in the picture on the left. And that's why we need to do the simplified ones. So we've talked about secondary structures and supersecondary structures. The next level would be a domain. And a domain is a compact substructure of a protein. It's the old story. I don't know what I can't define it, but I know what when I see it. It looks as if it would have independent stability. The one on the left clearly has a lower domain and an upper domain with a tenuous connection between them. The one in the center obviously has two lobes, and this is an enzyme that when a substrate binds, the two domains rotate with respect to each other to close over it like a clamshell. And the third is the one on the right is a fragment of fibronectin, and you can see the individual domains. There are actually a lot more. I've only shown 4 of them. And again, if you get bored with my talk, you can look at these pictures and try to pick out structural features. So on the lower right of the left-hand picture, you can see a helix pretty prominently. In the right-hand picture, the fibronectin, you can see beta sheets. So those are domains. These domains form a convenient way for proteins to evolve. Because individual domains, in some cases, have individual functions, but in many cases, the function of the whole protein depends on not only the composition in terms of domains, but how they're assembled. So here are pictures of a number of proteins involved in blood coagulation, and the pictures show individual domain structures. The full proteins on the lower right make use of, we say, mixing and matching these domains. They choose individual domains and string them along to form a protein. So all the proteins contain common domains as substructures, but the domains are organized in a different way in the different proteins. So they're making use of the same building blocks, but putting them together in different ways. And that's a very common mechanism of protein evolution. It's hard to make up a new protein from scratch. It's so much easier to reuse something that already exists, and evolution takes advantage of that. The next level, quaternary structure, involves the assembly of proteins from individual chains. Now, it's different from domains. If you have a multi-domain protein, it's one polypeptide chain. If you have hemoglobin, then it's 4 polypeptide chains. The difference is perhaps less than it appears, but it's been a classically recognized distinction. So myoglobin on the left is a single domain globin, and hemoglobin on the right contains 4 chains, each of which is related to myoglobin, and you can decide which of the hemoglobin chains is shown in the same orientation as the myoglobin chain. And once you've done that, you can ask whether you can see any structural differences between that subunit of hemoglobin and of myoglobin. Because these are related by evolution, and that's absolutely essential. Evolution is the clue to figuring out what's going on here. Because if you were to just show me a single protein and ask me what are the essential features, I have no idea. It's just too complicated. We let nature do the work for us. We look at proteins evolving, and we say, well, what nature has elected to retain, that's an important feature. What nature has thrown away, that's not an important feature. And that way we can try to answer such questions. Here is an example of 2 related proteins. lysozyme and alpha-lactalbumin, showing the sequence on the top and the structures at the bottom. Now, the sequences are aligned, and the letters in black underneath the alignment show the positions at which the residues are different. So most of the residues have changed, and yet the structures shown at the lower right are really still very similar. So evolution can push the sequences pretty far apart and still retain the structure. Now, if you showed me the two sequences and said, do they have the same structure? That would be a tricky question to answer. But seeing the structures, we could then go back to the sequence and say, yes, obviously they will have the same structure. I'm going to skip this for saving time. So, staying with evolution, the atomic event in protein evolution is a single base change. Some single base changes don't change the sequence or structure of the protein, but many do. And if we look at these aligned sequences, so we have human hemoglobin, alpha and beta chains. So hemoglobin's a tetramer. I showed you a picture. And it's a dimer of dimers. So there are two different chains in hemoglobin, 2 copies of each, so 4 altogether. And the 2 chains are called the alpha chain, the beta chain. And don't confuse alpha chain, beta chain with alpha helix, beta sheet. It's completely different. So the sequences shown are human hemoglobin, alpha and beta chain, horse hemoglobin, alpha and beta chain, and myoglobin. If we were to take the time to study these sequences carefully, we would discern that the closest relation between pairs of sequences are human alpha, horse alpha, and human alpha, horse beta. Next would be the alphas versus the betas, and then myoglobin's an outlier. So we can sort of see the course of evolution. First, the hemoglobin chains diverged from myoglobin, then they diverged from each other into alpha and beta, and then human and horse diverged from each other subsequently. And as the sequences diverge, the structures diverge. Now, if you just take a protein and make random mutations, there's a good chance you'll just destroy the protein completely. But if you restrict yourself to studying natural sequences and structures, that limits observations to things that you know exist and produce function. And usually then they maintain the same general structure. So here's 2 homologous related globins, sperm whale myoglobin and a globin from Paramecium. If you look at these, it's really not completely obvious what is preserved or not. They both bind heme, a small molecule called heme containing the iron, in the same place. You can sort of see that this helix corresponds to this one, and this one would correspond to that one. But look here, there's a helix there that doesn't correspond to anything. So how do we more precisely make quantitative the comparison of the structures? And the answer is you superpose them. Now, if you superpose them, you see that the heme groups fall on top of each other. The 2 sets of helices that I called your attention to are in similar but not exact positions. This is a kind of tricky picture to interpret. So let's see if I can get back. Oops. Well, I have a movie, but it's not showing up there, which is unfortunate. Oh, that's too bad. Nothing we can do, is there? Well, I better go on. If we can get back to it, we will. That's too bad. No, I'll just go on, okay, because otherwise people have to catch trains. The point of the movie is that if you can see it rotating around, the spatial relationships are much more clear, okay?
Can you close the open?
That was a mistake. There we go. So at least I'm sure you'll admit that you can see much more clearly what's similar about the structures and what's dissimilar about them, okay? I'll go back to the oops. Good, okay. Now, what we've seen in this slide in the movie is that there are parts of the structure that are similar and parts that aren't. And what we do is to extract the part that's similar. And this is called the core of the structure. So here what I've shown is a subset of the residues that appeared on the previous slide, but it shows only the parts that are well conserved. And that usually includes the binding site or the active site of an enzyme, and we call that the core of the structure. Here's another example. Spinach plasticyanin, cucumber stellocyanin, they're both copper-binding proteins, and this is the copper ion. Again, you can see similarities. Here's a beta sheet, here's a beta sheet. There's a helix here, not clear what it corresponds to. But again, to try to answer the question of what's conserved and what isn't, you do superposition. So here's the superposition showing everything. And here again is the extraction of the core, the similar bits. So you see that within these two structures, parts of it have diverged and parts of it have stayed the same. I'll give you one other example, which I prepared originally for a talk in Rome, but I love to show when I'm in Italy. There's an analog in the ceiling of the Sistine Chapel. So here, pardon me, there's a famous picture of God and Adam. If you walk down the room a bit, you see that there's a picture of Noah. And if you look at the pose of Adam and Noah, you see the lifted leg, the weight on the right arm. You say, oh, well, you know, this looks like hemoglobin and myoglobin. So if we're structural biologists, what would we do with these pictures?
Superpose them.
Yeah, you'd superpose them, right. And then you get something like this, which brings out the similarities. And one of the things you will notice, I actually made a movie of this, but I'm not going to try to show it. But this is the finger of God here, and this is the finger of one of Noah's sons. To a protein person, that would be like an aspartate side chain pointing into an active site achieving catalysis. But it further reinforces the idea that these were deliberately similar poses, not just accidental. And my idea is that Michelangelo was making a bitter joke that he said, look, you start out relaxed, listless, because you haven't been animated yet. God gives you life and all the magnificent opportunities that accrue, and what do you do with it? You go get drunk, and you reduce yourself back to the state that Adam was in before God brought him to life. Well, this is a digression, but anyway, that's my reading of these two pictures. So let me go on back to proteins. Oh, well, maybe. Yeah, okay, so what's my time? I'm not doing too badly. I'm now going to talk a little bit about what I and a few colleagues are doing in our research. If we want to understand how domains are put together, we have some ideas of what the substructures below the domain level are, and they include things like the supersecondary structures. The supersecondary structures account for a relatively small amount of the actual structures. So can you write down a complete parts list or equivalent of Lego bricks from which you can assemble all protein structures? In other words, can you dissect the structures into a library of pieces that give you a complete catalog of what the substructures are? There have been many attempts to do this, which accounts for our difficulty in getting the paper published. What would make one such set of substructures better than others? Well, you would have to ask how concise the set is. If I can give you 1,000 structures and you can give me 2,000 structures, then in principle, mine is more concise. Greater coverage. Can you really explain 100% of protein structure or 80% or what? And remember, the original identification of supersecondary structures was just anecdotal. People looking at pictures the way we've been looking at pictures and saying, oh, I see this, oh, I see that. Can you write an objective algorithm for that? Now, we've done it based on a representation of proteins that we call the Tableau representation. And the idea is that what defines a protein structure, if we compare myoglobin with plasticine and things like that, what defines a protein folding pattern is the set of secondary structure elements, the helices and stranded sheets, their contact patterns, and their geometric relationship. So we represent those data as a matrix or tableau, and interestingly enough, it's intelligible to both a human being and a computer, and it's suitable for algorithm design. So it's a matrix. This is acyl phosphatase. I've shown you this before. This is actually Annelise's structure. And there are 5 strands of sheet and 2 helices represented by beta and alpha, alpha A and alpha B, beta 1, 2, 3, 4, 5. And the rows and columns of the matrix are indexed by these secondary structure elements in the order of appearance in the chain. So if you were to follow the chain from N terminus to C terminus, you'd first go through a beta strand, then an alpha helix, then a beta strand, etc. The entries in the matrix are blank if the secondary structure elements do not make contact. So clearly the helix on the right and the strand last on the left are so far apart they don't make contact, and they would correspond to a blank in the matrix. On the other hand, the helices are packed against the sheet, so you would have entries. The entries are somewhat Kabbalistic, but what they are doing is recording the angle, the relative orientation of the 2 strands. And the argument is that this is what is the essence of this protein structure. The loops between them we don't really care about. And that's actually a long and very interesting story, which I will explore later this week. But in order to choose these substructures, we restrict ourselves to subtableaus. So this, for example, is a beta hairpin. It is 2 consecutive strands that are in contact, and if you interpreted the symbol, it would be that they're antiparallel. So we use subtableau, not necessarily consecutive rows and columns, as the elements of these Lego pieces. How to choose them? Well, we should describe the folding patterns concisely, provide greatest possible coverage, backbone only, no side chains involved. And the methods we use depend on a computer science technique called minimum message length criterion. This was a technique devised by Wallace and Bolton. It's a kind of algorithmic version of Occam's razor. You're searching for the most concise representation of the data set. And it's equivalent to the following scenario. There's a transmitter who wants to communicate information to a receiver, and canonically they're called Alice and Bob. It has to be lossless in the sense that the transmission of the information is complete and error-free. And Alice wants to encode the data such that she needs to send the minimum amount of information to the recipient. Now, suppose, for example, that Alice wants to send Bob coordinates of a protein containing alpha helices. Well, she could just send a list of coordinates. Or she could say, all right, here's coordinates of a standard alpha helix. Now all I have to send you is the position and orientation and length of the helices in this molecule. And that would produce a much shorter message. So the alpha helix would be part of a substructure library used to transmit coordinates of a protein structure. And by using such a substructure library, Alice will have reduced the total amount of information that has to be sent. Well, should she include supersecondary structures? So here's a helix hairpin. Is it more effective to send individual, to send a helix and then give the coordinates of the relative position and orientation of the 2 helices and the hairpin? Or should the hairpin itself form a separate substructure? It depends. It depends on how often hairpins occur in the data set you're trying to represent, and that's what the algorithm has to optimize for. It has to decide, well, should we just use a helix or should we use a helix hairpin? And using that, we used an unbiased data set of domains. Because the choices depend on how often things occur, you don't want to use the whole protein data bank, because the whole protein data bank has hundreds of myoglobin structures, and that would bias things. People have extracted unbiased, structurally unbiased data sets, and we determined an optimal library required to transmit all the folding patterns in this set using minimum message length. The result was 1,493 substructures. They vary in length. Shortest is 2, and the longest is 28 secondary structure elements. And it automatically included the standard supersecondary structures. We didn't put those in, they came out of the calculation. And then we went back and redissected the entire protein data bank, not just the training set, and that was the result. So here's a selection of the substructures, and they vary. This is actually, so this is, I think, I can't see. This is a tin barrel. But you see, some of them are quite big. Some of them look like whole domains or almost whole domains. Here's an example of the same library structure in related proteins and unrelated proteins. And notice that it fits much better in the related proteins than the unrelated proteins. But on the right, the two unrelated proteins containing the same library structure have the same sequence of secondary structure elements, making the same contact pattern with roughly the same orientation. There are a lot of potential applications of this. Some of the library structures are correlated very, very well with binding sites for particular ligands. So if you see such a substructure showing up in the dissection of a protein structure, you could be confident that, this binds the ligand associated with that. Some of the substructures give clues to function of the protein in which they appear. I should say as background that with all the genomic information that's coming out, the challenge has been, maybe you can even infer what the protein sequences are from the genome, although that's a long story. But given the structure of these proteins, it's still not obvious what the function is. And what the application of this would be correlation of dissection into substructures with function. Also, I showed you the prediction by Baker's group of a protein structure for an amino acid sequence. That approach makes use of local sequence structure correlations. And we think that the library dissection would be helpful in making those predictions. It opens up a lot of doors. And in principle, we could write down a complete periodic table of all possible protein structures. And then we'd be able to see what is observed and what hasn't been observed yet, and ask why. So maybe there are new principles of protein architecture that we don't know about that such an approach would reveal. If we make a complete table of protein structures, what's missing? Why are they missing? Is it physics? Is it history? You could test that by trying to design sequences that would fold into these things. And finally, where are we going? If you invite someone in 5 years to give a similar lecture, what would they say that I'm not able to say? The field is now in pretty good shape as far as understanding static aspects of isolated proteins. Biochemists have spent a century isolating proteins, studying their functional properties, solving their structures, and that's fine. What crystal structures, but not necessarily NMR structures, fail to reveal is protein dynamics. I mentioned that one of the multi-domain proteins had a conformational change. When it bound a ligand, it would close over it. You can't see that in an individual crystal structure, but that's something that is accessible to computing. So there's a whole field called molecular dynamics, which can reproduce some of these conformational changes. And in particular, even if you have a crystal structure of this enzyme in the open form and a ligated crystal structure in the closed form, there's the question of the pathway. And the crystals and even NMR structures don't tell you the pathway, but computing can. And there are a number of very interesting papers on how proteins change conformation. Now, the main thing is the sets of interactions. So individual proteins don't tell you how cells work. There are protein interaction networks that involve physical interactions between proteins. There are logical regulatory networks that show how the whole process is organized, and that's the current challenge. So for a century, biochemists have been taking cells apart, and our job now is to put them back together. So that's what I wanted to say, and I'd be glad to answer any questions, if I can, that you have.
Thank you very much for your very exciting lecture. Here, first question.
Thank you for the very nice lecture. I have one question. You said that you look at changes in protein along their natural evolution. And how do you tag the natural evolution of a given protein?
Well, there are some cases that are obvious. So if you take human hemoglobin and horse hemoglobin, they have very similar structures, very similar sequences, and they have the same function in both species. So you can be fairly confident that they've diverged from a common ancestor. In other cases, it's not easy to tell. Annalisa and I spent a year and a half looking at the globins and phycocyanins. So globins are oxygen storage and transport proteins in mammals and other vertebrates. Phycocyanins are accessory pigments in chloroplasts that absorb light in regions that chlorophyll doesn't absorb and transfers the energy to chlorophyll. Now, when Robert Huber and Tilman Schermer first solved the structure, they were amazed to see that it looked similar to the globins. But here you have a completely different function. The structures were certainly much less similar than human and horse hemoglobin, or even myoglobin and the paramecium globin that I showed you. And it was certainly not obvious whether they were related or not. And Annalisa and I spent 18 months trying to resolve this, and we finally came down on the side that they were related. But it was certainly not obvious, and you know, we had to really take the structure apart, atom by atom, to decide.
I would like to ask you about, in one of your last slides, you had, from this library, when you have it complete, what is missing? And is it physics? Is it history? So, well, I would say it depends on how large is the size of your elements, because the space of protein shapes has certainly not been occupied during evolution. It is occupied by a very, very small amount of all the possibilities. So, I mean, I would guess.
You may well be right. And in fact, the very interesting paper that points out that when we get the structures of all the proteins of the viruses in the ocean, we may find 10 times as many protein folding patterns as we know now. So you may very well be right. I'm not saying you're wrong, but I would sort of like to find out. And the point is, I showed you the definition of the tableau. It's child's play to write down all the possible tableaus. But here's the problem. They're not all physically realizable. So if you were to write down a tableau that contained 3 alpha helices, all pointing mutually in opposite directions, you can't do that, because once you have 2 antiparallel alpha helices, the third one must be parallel to one of them. So asking that you find a third helix antiparallel to both of them is physically not realizable. And to eliminate the physically unrealizable tableau has been a very difficult technical problem.
Yes, thank you for the lecture. So I have a very simple question. So you mentioned that actually once you determine the sequence of the amino acid, then they know how to fold, essentially.
They
so,
know how to
Yeah, they know it. So, and I understand that for us it's very difficult to understand how they will fall. But what is the physical force that is driving the folding of the sequence? Is it, you know, chemical bonds or some van der Waals forces? I don't know.
Suppose, for example, you take a protein and you heat it up so that you break down the structure. You've got this linear chain, and it's got all kinds of conformational freedom. It doesn't have a unique structure, but it's flopping around. Now, every time you bring two parts of the chain together, then you have amino acids interacting. And those interactions can either be favorable, they could form hydrogen bonds, or you could have a positive and a negative one coming together, or they could be unfavorable. And what the protein is doing is maximizing the amount of favorable interactions. And in fact, they've evolved so that there is, in most cases, a unique one. If you were to just synthesize random polypeptide chains, that wouldn't be true. You wouldn't get a folded state. But you've evolved a case in which there's one organization, spatial organization of the chain that has a maximally favorable interaction, and that's your native state. Now, how the protein finds it, that's another question, and many people believe that it's a hierarchical process, so that you might first form alpha helices, and then they could come together. So again, there's an important difference between the fact that the alpha helix is a local structure, and a beta sheet could bring together two strands from far apart. That would be harder to find from the denatured state. And it also looks as if the proteins have evolved pathways of folding. Those are more obscure because it's difficult to describe them. And given that you're starting from a large number of possible denatured conformations, basically each of them has a trajectory to folding. So it's not even a unique answer. So that's a difficult question. People are working on it. And there are some general ideas, but it's not an easy question to answer.
Okay, thank you. Thank you, Arthur, for this great talk. Also very, very clear, also to people like me who come from a completely different field. In my case, it's linguistics. So I would like to say that I was impressed by the similarity between your perspective and the linguistics perspective, coming from a superordinate level, including the interaction between physics and chemistry with historical dimensions, to a more specific details, points like the relationship between information, structure, and function, combinatorial processes, the role of core elements, etc. In particular, I would like to focus on the protein structures that strikingly parallel linguistic structures, including contact patterns and geometrical relationships. So I'm wondering, I'm actually convinced of this, but I'm asking you for an opinion concretely. I'm wondering whether linguistics and biochemistry and molecular biology and all these related disciplines can benefit from each other on this specific topic. So to understand protein structures, how protein changes, and in order to understand how this cell, the mechanisms undergoing the combination of these cells. You conclude your talk saying that the challenge is to put them together. So I was thinking about it. How could we apply the linguistic methods to address this issue in molecular biology, and vice versa? How linguists could benefit from the molecular biology perspective to understand not only syntax, because that's clearly syntax. While you were giving your speech, I was thinking, I very often use the same same sentences, same words, same expressions to explain to the students many linguistic phenomena. So how could we also apply your methods to linguistics to understand not only syntax but also morphosyntax, but also in particular, that's one of my main interests, the relationship between structure, formation, and function. Also diachronically, that in your term is evolutionary. Thank you, Arthur, for your great talk.
Well, that's a very, very large subject. I mean, I once wrote that the Romance languages were the Darwin's finches of linguistics, so maybe it should be the other way around. And by the way, there have been people, David Searles was one who really tried to push this analogy. My basic problem with it is that, to me, language, whether written or spoken, is basically one-dimensional. Sorry. And protein structures, life in general, is three-dimensional. And how you jump that gap is the tricky bit.
It's not one-dimensional, by the way.
Oh, all right. Well, then I'm wrong. Good. I learned something then.
Right. You have to think, also, that was another detail where I noticed the similarity. So, three-dimensional, not only diachronically, space, time, socio-historical levels, but also you can analyze a sentence in more than one dimension, including formal and functional components. So at least you have to think about the fact that when you say something, when you use the language, at least you always have the main issue is to encode in linguistic structures, discrete linguistic structures, conceptual representation. So at least you have two dimensions on the table, but these two dimensions cross by many other dimensions to solve the issue of encode into discrete structures, multifactorial and scalar concepts. So the pluridimensionality is another issue in language, is a fundamental issue. So we could really, I guess, if you think it's something concretely pursuable, we can think about it. Thank you.
Well, it's a very interesting idea. I must say my ignorance doesn't allow me to get very far with it, but I'm sure people could.
Very nice talk. I was just wondering if, so the basis sets that you discussed, the structure that you use to decompose the proteins, is it obvious, like in 100% of the cases, that something like an alpha helix is only described, characterized by, I think you said 3 numbers, orientation, position, and length, or something like this? Or are there deviations from this? And if so, like a source of noise, if so, can you characterize, is there a physically motivated way of characterizing deviations from this?
No, there certainly are deviations. They're usually pretty small, but not always. But for lossless information transmission, Alice would have to end up with Bob knowing exactly where the atoms are, okay? Now, the point is that even if the helix shows deviations, it's still more efficient to send, oh, this is a helix, and now I'm going to send you the deviations. Because the smaller the numbers you send, the less information you need. So the strategy would be to say, there's a helix at a certain position, orientation, and space, but there are deviations from it, which I will now send you, and the hope is that that's the most efficient way to send it.
Characterize deviations with some other basis sets, presumably.
Well, usually it's just, again, if you want to be as clever as possible in encoding what you send. So far, our approach has been you would send different delta X, delta Y, delta Z, and that would be it. There might be a better way to do it, but.
There's one more question. Yes, a more of a general setup. I was wondering whether reinforcement learning was applied to this general problem with some strict setup, and in case of yes, the advances of this? Reinforcement learning is the trial and error technique.
I don't know, maybe Rita's better at answering that than I would be.
Sorry?
What kind of learning? I'm not sure.
Reinforcement learning.
By computer programs?
Oh, yes.
Well, the whole field of machine learning and artificial intelligence applied to these problems is a very big one. And as I say, Rita is one of the world's experts in that. It's not something something I've read about, but that I've never actually done it. But there's an immense literature on it. I mean, look up Casadillo in PubMed. Okay. You'll find lots of papers.
Thank you. So anyway, Arthur will stay now with us in a small refreshment. So whoever has more questions is welcome to ask them directly. And I thank very much our distinguished speaker for a very talk, as was witnessed by the many questions from a diverse audience.
If anyone wants to see the other movie, we can put it up here and just come look at it. It's sort of fun.
Okay, thank you very much.
Thank you. Why isn't the mouse right?